Vitamin B6 is a colloquial umbrella term. It does not refer to a single chemical molecule. It represents a family of three distinct pyrimidine derivatives: pyridoxine, pyridoxal, and pyridoxamine. These precursors have diverse dietary origins, sourced from fish and meat to legumes and oil-rich seeds. Once ingested, the human body subjects them to chemical alteration. This metabolic alteration funnels all three variants into one singular coenzyme, the active form, pyridoxal 5-phosphate, or PLP. The biological mandate of this molecule is extensive. PLP functions as the obligatory coenzyme for over 140 distinct enzymatic reactions throughout the body. By converting dietary precursors into this single coenzyme, human physiology relies on PLP as a central hub of cellular metabolism. It is a critical chemical choke point. Establishing that hub requires a foundational phase of biosynthesis and activation. First, the dietary B6 variants are phosphorylated by the enzyme pyridoxal kinase. Second, these newly phosphorylated compounds are oxidized via the enzyme PNP oxidase. This two-step kinase and oxidase pathway is the biological gateway required to yield active PLP. From this active center, the metabolic network expands outward to its first major domain, amino acid metabolism, operating specifically through transaminases. Transamination is the biological process where a substrate amino acid is converted into a keto acid. For this conversion to succeed, an amino group must be extracted and transferred. PLP actively steps in during catalysis to capture this amino group. By capturing the amino group, PLP alters its own structure, transforming into pyridoxamine phosphate to facilitate the reaction. PLP operates as an active molecular shuttle, driving the mechanics of transamination from the inside. The network branches further into the major synthesis cascades, beginning with the production of blood components through delta-aminolevulinic acid synthase, or ALA synthase. This is the master enzyme for heme synthesis. It executes the rate-limiting step of converting succinyl-CoA and glycine into delta-aminolevulinic acid. To catalyze this foundational step of heme production, ALA synthase strictly requires PLP as its coenzyme. The network also governs the endogenous production of vitamin B3, known as niacin. Rather than relying on dietary intake, the body synthesizes its own niacin starting from tryptophan. This multi-stage transformation funnels down to one critical final enzymatic step. The enzyme kynurenase drives this step, and its catalytic ability entirely depends on PLP. Without the central PLP hub, the endogenous production of both vital blood-carrying molecules and essential vitamin B3 comes to a halt. Extending into neurological functions, PLP dictates the biosynthesis of major neurotransmitters. The biosynthetic pathways creating dopamine, adrenaline, and serotonin all require PLP-dependent enzymes to function. The final biochemical mechanism on our network map is glycolysis, operating specifically within glucose metabolism. The body utilizes the enzyme glycogen phosphorylase as the primary catalyst to break down stored glycogen. PLP actively participates in this catalytic process, directly linking vitamin B6 to glucose mobilization. PLP connects glucose mobilization, brain chemistry, and blood synthesis into a single metabolic requirement. By deconstructing this network map, we can correlate the biochemical failures of specific enzymes directly to macroscopic clinical pathologies. If the central PLP node is removed, ALA synthase fails. Heme isn't produced, resulting in an anemic state. Further down the map, kynurenase fails. This creates a cross-vitamin dependency. Without niacin synthesis, a B6 deficiency triggers a B3 deficiency, manifesting as pellagra. Finally, dropping neurotransmitter synthesis creates a synaptic imbalance, leading to peripheral neuritis. A single vitamin deficiency produces diverse symptoms across blood, skin, and nervous systems, representing the systemic failure of a central enzymatic hub. To maintain this network, the baseline daily requirement of vitamin B6 is established at 1 to 2 milligrams. During periods of increased metabolic demand, such as lactation or pregnancy, daily intake must rise to 2.5 milligrams. However, vitamin B6 presents a specific clinical risk during oversupplementation. While a deficiency causes systemic breakdown, excessive intake leads to clinical toxicity. The macroscopic symptoms of B6 toxicity manifest as neurological degradation, specifically resulting in nerve damage, numbness, and muscle atrophy. Because PLP acts as a universal coenzyme dictating over 140 chemical reactions, precise biochemical homeostasis of vitamin B6 is mandatory for healthy human physiology.